Good evening, everybody. Am I audible? You can use the chat window. So I'm going to share my screen. Let me know if that's visible and you can hear my voice. Okay. So you can see my PPT, right? Okay, so we couldn't hold the session yesterday. I was not feeling well, therefore, I couldn't hold the session on Friday between 6 to 8 as we usually do. So there's something I need to change. All right. So we'll start. I welcome you all to the seventh week of live interaction sessions for the course Bioinformatics, Algorithms and Applications. So I'm Lavanya N, a PhD scholar at IIT Madras, who will be helping you through these live interaction sessions, usually on Fridays between 6 to 8 p.m. So this week's practice questions will be focusing on protein structure analysis part 4, protein structure prediction 1 and 2, and protein stability parts 1 and 2. Looking into the first question, pairs of cysteines within a distance of dash angstrom between sulfur atoms are considered as disulfide bridges. Option A 4 to 5, option B 2 to 3, C 5 to 6, and D 6 to 7. Okay, Debangana has answered. What about the others? I'm going to put this in presentation mode. Thank you, Bala. The distance between somebody has to mute themselves. Venkatesh, will you please mute yourself? Sorry about that, people. Getting back into the question. So last week we had seen that 5.3 angstrom is required for sulfur aromatic interactions, right? So between sulfur atoms, the minimum distance required or the cutoff distance is generally maintained between 2 to 3 angstrom, right? Let's move on to the next question. Which of the following atoms can form a hydrogen bond? A. Sodium, B. Oxygen, C. Aluminium, and D. Rubidium. Thank you, Vijayanta. Others? Okay, good. So I see three votes for oxygen. Anybody else wants to take a guess? All right. So I have here the electron structure distribution of sodium, oxygen, aluminum, and rubidium. You see that only oxygen is electronegative enough to form a hydrogen bond, right? All the others have three here. There's only one in the outer orbital, and again one here, right? But oxygen has six, and it's just wanting two to complete its shell. Therefore, oxygen is the most electronegative among the options here. All right. Question 3. Regions of proteins which have similar structures are called A. Structurally distant regions, B. Structurally conserved regions, C. Structurally distort regions, and D. Structurally identical regions. Okay. Vijayanta has answered again. Anybody else? So one vote for conserved. Distant and distort seem off, right? In having similar structures, it's not going to be distant and definitely not distort. So the options that you should consider are conserved and identical. Bala has voted for identical. Anybody else? So given multiple proteins, regions that have A similar structure is just a simple representation, and others might differ a little. But in this particular region, these group of residues always seem to form a beta sheet or beta, uh, or say a alpha helix is said to have not identical but conserved. Right, you can call these regions as conserved regions where amongst different organisms at various levels you're seeing the same structure being conserved, but you cannot say they are identical. Some minor changes can always be there, but they can be similar. Similar does not necessarily mean identical, right? So option B is the right answer. Question 4. Which experiment stated that the structure of a protein is determined by its amino sequence? Option A. Anfinsen experiment. B. Circular dichroism. C. Nirenberg and Mattei experiment. And D. X-ray diffraction. Vijayanta says X-ray. Take a wild guess. Ashira also says X-ray. So let me make that 2. Bala also agrees with 2. So why not the others? So look into the question again. Which experiment stated that structure of a protein is determined by its amino acid sequence? So it is the sequence or the chain of amino acid, and what comes after what determines the structure. So circular dichroism and X-ray diffraction are techniques. Circular dichroism will give you an idea of the secondary structures of a protein, or it can give you an idea of the tertiary environment in a protein, right? But does it tell you anything about a structure of a protein being determined by its amino acid sequence? No, right? Same is the case with X-ray diffraction. It's an excellent technique to obtain high resolution protein structures, but it gives you, or it does not prove anything that the structure of a protein is determined by its amino acid sequence. So this leaves you with 2 clues. It's either A or C. And those who have already answered can also attempt again. No googling in between. So if you have previously done some course on genetic engineering or molecular biology, you would have definitely come across Marshall Nirenberg. Okay, Savinder has said C. Maybe let's take a minute until we get more answers. So this was an experiment where they said that 64 codons are present, but only 20 amino acids are known, right? And they recognized three codons that stop the synthesis of proteins, or also called stop codons. Still, you have 61 codons for 20 to code for 20 amino acids, which means more than one or two codons code for the same amino acid. So the degeneracy in codons was found by Nirenberg and Mattei. However, this question therefore leaves only A to find out. If you directly know or aware of Anfinsen experiment, you can straight away shoot at A. Right, so here I have the answer as A, and the following slide. um, yeah. So this is the Anfinsen experiment. He was given a Nobel Prize in chemistry. So I just have tiny excerpts. I did not put the link for this part, but there's a link for the second part. Right, so problem of relationship between structure and function in enzymes. So on the basis of studies on ribonuclease, so he proposed the information determining this tertiary structure of a protein resides in the chemistry of its amino acid sequence. So this is the entire question or concept about. Okay, he did multiple experiments, and he saw that many proteins could spontaneously refold to their native forms even after this cleavage of disulfide bonds and disruption of the tertiary structure. They could still refold into their native forms. So there is something about the sequence of amino acids that allow it to fold in a certain manner, right? So you can read about this later. And this was Nirenberg experiment. So Nirenberg and Mattei selected E. coli bacteria cells as a source of cytoplasm. So these guys, they did an interesting experiment where they designed experiments to determine how DNA works free of complicated biological process. So in a cell-free system, when when RNA is present and all the ribosomes are there, and if you add different amino acids, so they added E. coli extract to 20 test tubes, each containing a mixture of all 20 amino acids. In each test tube, one amino acid was radioactively tagged. So they had 20 different reactions where in each you had, say, glycine was radioactive, alanine was radioactive. So radioactively labeled all amino acids in each of the tubes. Only one of them will be labeled. All the others are normal amino acids. And they found out how the first breakthrough was use of poly-U uracil, and as a model, they identified the nucleotide combinations for incorporation of other amino acids. And they also found that the coding units for amino acid contains three nucleotides or triplet codon. All right, and combining four nucleotides in a 3-liter code yielded 64 possible combinations. So sufficient to describe 20 amino acids. So four nucleotides AUCG, because we are talking about RNAs here. And if you put it into three different all possible combinations, if I have AAA, AUA, AGC, and so on, you'll get 64 such combinations, and that's how they figured out the amino acid codons, or how they are coded. So the answer for the previous question is Anfinsen's experiment. Question 5. In protein 3D structure prediction, model optimization is used to option A, remove atomic clashes, B, minimize the potential energy of the structure, C, reduce torsional angles in disallowed regions of Ramachandran plot, and D, all of the above. So among the many steps of model prediction or protein 3D structure prediction, model optimization is the penultimate step. So think about the options. Is it to remove atomic clashes, minimize the potential energy of the structure, reduce torsional angles in disallowed regions of the Ramachandran plot? Thank you, Ashira. One vote for D. One more. Great. So, can we all agree that at this stage of model optimization, all three processes, removing atomic clashes, minimizing the potential energy of the entire structure, and reduce the torsional angles in disallowed regions of Ramachandran plot, are all part of model optimization? Right? So, the correct answer is all of the above. Which pair of cysteines form a disulfide bond in the following? So initially we had seen that you need 2 to 3 angstrom distance for a disulfide bond to form. So the options given are option A cysteine 2 and 4, option B 1 and 2, option C 1 and 3, and option D 3 and 4. Shira says 1 and 2 and 4. Others, Debangana says 3. Vijayanta goes with A. That's it. So how do you calculate the distance? In a three-dimensional structure or in three-dimensional space, if there are 2 points, how do you find the distance between these 2 points? Right? And this has to give you an answer between 2 to 3. Let's keep it as 3. Let's round it off to 3. Right? So ideally x1 minus x2 the whole square plus y1 minus y2 the whole square plus z1 minus z2 the whole square should give you a maximum value of 9. Agreed? So let's look into these pairs. First is 2 and 4. So 2 is here and 4 is here. Let's. I'm going to use a calculator here if required. But let's just go with the face values. 4 and 4 is going to hardly give you a point 0.5 square for y. And here I'm seeing another 1.3 maybe. Yeah, this is 1.6 and probably 1.2 here, right? And between these you have 3.5, let's say. Okay, so leave these. So here this will give you 1.2 and 2 square will be, I guess, 1.44 plus 0.25 plus I don't know what 35 square is, but definitely it's going to be over 9. 3.5 square is 12.25. So this is greater than 9, right? So the cutoff distance is more. Option A is gone. So can you work on this similarly and then choose an option? So you have B, C, and D to choose from. I'll be back in a moment. Answers? Okay, let's do this together. Yes, Vijayendra. So he also goes with, oopsie, C, but I guess you guys got a glimpse of the answer. So let's first check C. 1 and 3. So between 1 and 3, it's hardly zero point 0.8 square plus here you have some 0.9 square plus this will be about, let's say, 1.7 square. So 0.64, 0.81, and this is 2.89. So whatever uh, the sum is, it's going to be less than 9. So this qualifies. So C at least qualifies. What about 1 and 2? You have a major difference here. 16 square itself has done the job, so can't be. What about 3 and 4? Again, you have a major difference of 17 square here, so it's gone. So C is your answer, as Devangana rightly pointed out and Vijayanta eventually realized. All right. Question 7. You guys can hear me, right? Alright. The concept of protein folding problem is based on the notion that a amino acid sequence of a protein contains the information about its 3D structure. B amino acid sequence dictates protein folding. An amino acid sequence is sufficient for determining the fold. D all of the above. Thank you, Vijayanta. Others? Okay. Shira goes with C. Anna goes with D. Any takers for A or B? Alright, so the protein folding problem is the gap between the number of sequences available and the structures that are predicted or found, determined by experimental methods or computational means. And still, how there's a huge gap between the number of sequences available and the number of structures available. So, the protein folding problem is to determine more structures from the amino acid sequence. So, if you see the options here of a protein, amino acid sequence of a protein contains information about its 3D structure, which was the basis of protein folding problem. So, is that true? Yes. So, amino acid sequence dictates protein folding. Yes, if the sequence was different, although different amino acid sequences could give rise to a same protein fold, the problem is based on the notion that if you have conserved regions or amino acid sequences between different proteins, it is more likely that they will adopt a similar fold. Right? So, this is also true. Amino acid sequence is sufficient for determining the fold. Again, true. So, all these notions were the basis for the protein folding problem. Therefore, option D is right. Question 8. Ab initio modeling is a physics based approach, B. Knowledge based approach, C. Homology based approach, and D. All of the above. Thank you, Debangana. One vote for A. Bala says B. So you agree that it's not homology or all of the above? Just two options. Vijayanta agrees with A. Ab initio modeling is done when you have no predecessors or no homologous protein available, right? So this knowledge is not helping us much, but the sequence is the sole source of information for us now. And we are starting with the first amino acid and attaching the second amino acid based on the bond length, third with the angle and the bond length, and second with just the length, and third with angle and length. And when you go to the fourth, you have length, angle, and the torsional angles here. It's phi or psi, right? So by residue we keep continuing. Again you will have all four. Here again you'll have length, and this is for fourth, right? So when you go to fifth, you will have length as well as angle as well as torsional angles, and so on. So it's a physics based approach rather than knowledge based, and definitely not homologous. All right. Question 9. Atomic solvation parameters of n plus and O minus atoms are: option A positive and positive, B positive and negative, C negative and positive, D negative and negative. I don't know if you all remember the values of n plus and O minus from your lectures. If you have made notes, yes, or in case you remember, great. But think about these. The solvation is, what is it for polar and non-polar? So I hope you remember this sigma, which was the atomic solvation parameter, and there were five categories. And we are talking about hydrophobic free energy values. You can take a random guess, there's no harm. C, and there was N or O, N plus, O minus, and S. Some of these were positive and the others were negative. Take a random guess. Vijayanta goes with D, Ashira with C. Thank you, Ashira. Okay, Bala. So even if you don't remember the exact values for each of these atoms, I'm going to write down the exact values here. It was for C 12.02 calories per mole Armstrong square, and for N or O it was minus 5.86. For N plus it was minus 19.46. O minus is minus 34.98, and S is 35.51, and the units hold calories per mole Armstrong square. So for Bala, the atomic solvation parameter is a part of determining the hydrophobic free energy of atoms or proteins. So is given by hydrophobic free energy is sigma I into delta accessible surface area of I, right? And this delta ASA can be expanded as the surface area, solvent accessible surface area of folded state minus solvent accessible surface area of unfolded or extended state. So how do you find these? The solvent accessible surface area of folded state can be found from the 3D structure in PDBs, so you can get the information about how much the surface area is accessible. For unfolded or extended state, you use the values for a free amino acid residue, or when you have the glycine X glycine. So this is the any amino acid that is for which you need to find the accessible surface area in an unfolded or extended state. Now what about this sigma? This is the atomic solvation parameter. So when you see, it is positive for hydrophobic or non-polar atoms and negative for polar and charged atoms. Now, how is this sigma found? So this was explained as finding the value for each of the 20 amino acids as a matrix format, and for each of the different atoms C, N or O, N plus, O minus. So here they have assumed N and O to have similar solvation uh, parameter, and you have these as a 5 cross 1. Again it will be 1 2 3 4 5. So by looking over multiple structures, they have arrived at these empirical values for C, NO, N plus, O minus, and S. And if you're asking for the physical meaning of what it means, it is more about will it solvate with the solvent in the solution or stay in the hydrophobic core, right? So we saw about whether the transfer is favorable or unfavorable based on whether it is hydrophobic or hydrophilic, how it partitions between two different fluid conditions, polar and non-polar solvents. And this forms a part of the equation for finding free energy due to hydrophobic interactions. I hope this was clarifying enough to start with. In case you need more detail, I'll have to attach a bit more of an extra slide for this. So going with these values, N plus has minus 19.46 and O minus has minus 34.98, meaning both are negative. Option D is the right answer. Question 10. 3D structure models of a protein obtained with homology modeling using a template with 90% sequence homology is similar to option A high resolution X-ray structures, option B low resolution X-ray structure, option C high resolution NMR structures, and D not comparable with experimental structures. Bala says A. Others? So 3D structure models of a protein obtained with homology modeling and template with 90% sequence homology. So how would such a structure be? Vijayanta says C. What about B and D? Shira goes with C. See, although the template sequence homology is 90%, you can never compete with a high resolution experimental technique, right? So this throws both A and C out of the picture. And not comparable with experimental structures is too on the other side, right? Both are extremes. You cannot compete with a high resolution X-ray structure, or you can neither say a homology modeled structure with template with 90% sequence homology will be bad, so bad that you cannot compare with experimental structure. So your answer would be B, where it is similar to a low resolution X-ray structure, right? So let's quickly go through week seven's assignment. I think you are all aware of the answers and have already finished and submitted the assignment. Question 11. Comparing solid, liquid, and gas, which medium has the minimum entropy? A. Solid, B. Liquid, C. Gas, D. All the states have the same entropy. Jayanta says solid. Others? Thank you, Ashira. Cool, right? Which has the minimum entropy? Solid. Which has the maximum entropy? Gas. Right? So option A is the right answer. In homology modeling, if 100 residues are aligned with 30% sequence identity, which zone does it fall? Twilight, safe, perfect, dangerous. Okay, Vijayanta and Ashira say twilight. So it's just a rough diagram. So let's say 30 falls somewhere here and 100 falls somewhere here. What was it? 50 and 100. You can explore the chart, but it's not the safe region. It actually falls somewhere in the twilight zone. Definitely not safe. You will need at least some 60% to fall in the safe region. My axis is a little distorted, so it should probably be here. But it falls in the twilight zone. Not perfect, not dangerous, not safe. Question 13. The free energy change between folded and unfolded states of a protein is in the range of 0 to 5 kilocalories per mole. Option B, 5 to 25 kilocalories per mole. C, 50 to 100 kilocalories per mole. And D, 100 to 500 kilocalories per mole. Thank you, Vijayanta and Ashira. I think this was a very straightforward, also Bala, question or answer given in the videos. So there's no doubt about it being 5 to 25 kilocalories per mole. Question 14. Which of the following is dominant in the unfolded state of a protein? Okay, Vijayanta says D. Is it A, hydrophobic, B, van der Waal, C, electrostatic, and D, entropy? Venkatesh says D. All the other options A, B, and C, hydrophobic van der Waals and electrostatic forces of attraction, are contributing towards the enthalpy of the protein, which dominates in folded state. Right? So entropy is the only option that it dominates the unfolded state of a protein, and therefore D is the answer. Question 15. If NH bonding with O forms hydrogen bond, identify the hydrogen bond donor. Is it N, H bound to O, or O, or NH? This is a hydrogen bond interaction. Vijayanta and Ashira go with A. So does Bala. So obviously, right? So if the bonding is going to happen between H and O, the molecule, or sorry, the atom that is attached to the other end of H or covalently bound to H needs to be electronegative, and it has to or partial negative character, which will snatch electrons or distort the cloud so that there's a partial positive character with H, which allows it to form a bond with O. So the hydrogen bond donor is N. Question 16. In backbone generation, if the template and target residues are identical, option A, all main chain and side chain atom coordinates are copied. The main chain and side chain coordinates of the same amino acids are copied. Only main chain atoms coordinates are copied. Only side chain coordinates of the same amino acids are copied. One note from Vijayanta. As well as Ashira. Bala says B. Vijayanta says, but the right answer is B. So when I saw this question, let's wait for others to answer too. Any takers for C or D? Or shall we rule it out straight away? So like Vijayanta I was and Ashira, I was conflicted between is it A or B, right? But look at the question. In backbone generation, if the template and target residues are identical, we are not talking about the entire stretch of sequences being identical. If a particular residue is identical, what should I do? The main chain and side chain atom coordinates are copied, or main chain and side chain coordinates of the same amino acids are copied. Both answers are very similar and very close to correct. But if you see the specificity, the main chain and side chain coordinates of the same amino acids are copied. I would argue that option A also satisfies the question, because if the question says in backbone generation, if the template and target residues are identical, they have not even said anything like similar. If you go by which option is specific, I would say B. Although, and if it had been sequences, then B makes even more perfect sense since they've spoken about if the template and target residues are identical, and if you look at option A and argue that these are, why can't I copy everything, then essentially you're not doing anything, right? You have a target and you have a template. If they are absolutely identical in everything, and if you copy all the residues 100% identical, then you can copy the backbone as well as the side chain coordinates, and therefore you have just copied the template structure. There is no model generation at all, there is no backbone generation, and therefore I'm siding with B and not A. Does that solve your question, Vijayanta? So the main chain and side chain coordinates of the same amino acids are copied. Okay. Question 17. Van der Waals interaction between atoms I and J are defined as. So what is the formula for Lennard-Jones potential? Is it summation of Aij by Rij power 12 minus Bij by Rij power 6, or the signs just differ? Plus and minus here, minus and minus, plus and minus. So which combination is right? I think Vijayanta and Bala have said A. Which is indeed the answer, right? There's nothing much to think here. Option A is the right answer. Question 18. CASP stands for Critical Assessment of Protein Structure Prediction, Crucial Assessment of Protein Structure Prediction, Critical Assessment of Protein Sequence, and D. Clustered Assessment of Protein Structure Prediction. Clustered, critical, crucial. Assessment is same, of protein same. Right? Structure prediction, sequence, structure prediction. So critical is right, structure prediction is right. So no other option has this combination except for A. Critical assessment of protein structure prediction. That gives answer A. Thank you, Vijayanta and Ashira. Question 19. Consider atomic mass for C, N, and O as 12, 14, and 16 respectively. What is the center of mass for glycine with the following X, Y, and Z coordinates? Option A 6.8, 3.0, 9.0, B 6.8, 2.8, and 8.0, C 7.1, 2.1, and 7.8, D 6.5, 2.5, and 6.2. Vijayanta says A. Any other answers? And how do you calculate this? Center of mass. The answers are given in X, Y, and Z coordinates. That's right. center of mass, summation of mass into coordinate mi, right? Similarly, you will do for y and how do you solve this? Let's take x. So how should I sum it? So Cm, the x coordinate would be, what is the mass? First let's do nitrogen, 14 into 6.3 plus carbon, it is 12 into 6.1 plus again 12 into 7.4 plus 16 into 7.4 divided by 14 plus 12 is 12 plus 16. So that would be 14 into 6.3 plus 12 into 6.1 plus 7.4 plus 16 into 7.4. So this gives 368.6 divided by 12 to 24, 38, 44, 54. So the answer is 6.82. This leaves us with option A and B as the probable answers. Now when you do for Cm of y, what do you get? The denominator is going to remain the same, 54. So I'm going to have 14 into, look at this now, 4.5 plus 12 into 3.5 plus 2.8, because both of these are carbon, plus 16 into 1.6. So what does that give? 14 into 4.5 plus 12 into 3.5 plus 2.8 plus 16, which is 164.2 divided by 54, 3.04. So this is for people who still haven't figured out how to do this. For others, I guess you're good to go. So this gives 486.2 divided by 54, which is 9.003. Vijayanta, it's not 42, rather 54, because you're doing two carbon, right? So I have to sum both. If I were doing 42, then you will not get any of these, right? You're doing four atoms, each of this mi. So m of n plus m of c alpha plus m of c plus m of o. So all four will come into, how many ever nitrogens you have, you will add all of those. So if it's 12, you will have 2 into 12, because there are two such carbon atoms in the denominator, and you are also doing two such additions the numerator as well, right? So with these calculations, you know that the answers are 6.82, 3.04, 9. So closest option would be A. And the denominator is 14 plus 12 plus 12 plus 16. It's not 42, it is 54. Question 20. The principle used in structure comparison with DALI server is hydrophobicity, contact map, radius of variation, and sequence alignment. Vijendra says B. Others? Shira says B. Bala says B. Also Debangana. The right answer is contact map, right? The DALI server goes with contact map. We had seen a picture of the server in previous week or a week before, I think. So I didn't put in any uh, justification for this answer because I thought it was a straightforward answer. But I can include something while I post the PPT. Alright, so the answer is P, contact map. So with that, the questions for this week are over from my side. In case you want to tell me something, you can drop in a text. Otherwise, we can close this session. Thank you all for attending because I was not well and I did not expect you all to join today to continue with the same TA. So, thank you all for attending today's session. Yes, Venkatesh. If there's some question that you want me to repeat, also you can do that. Thank you, Bala. Thank you, Jayanta. Venkatesh, I don't think. Thank you, Surinder. That's a very vague question. Tell me through research topics. If there's anything else connected to this week's session, um, first of all, it's beyond the scope of this session. And second, I'm not qualified enough to give you research topics on the things that you just mentioned. I'm actually from a protein biophysics lab, and my expertise in bioinformatics is very limited. So, computational biology, you should take this up with somebody else. And there are so many research topics. You cannot say this is the major ongoing or hot cake right now. You can always read more literature, surf more things, look into the journal publications. There are always highlights in most journals, right? So you can see and find what's ongoing. You can start with science, cell, nature, nucleic acid research. We saw different methods to create non-redundant data sets, right? You can refer the previous lectures or videos, CD hit, for example. There are different libraries available. You have to, I mean, with bioinformatics, you can search a lot, you can find a lot. The whole point of informatics is use internet, use all the available information, and you can find a lot by yourself. Yes, there's a website in IIT Madras biotech department. You can find that. Great. Okay. Uh, you can restrict because I think others will be lost in this conversation. You can always mail me if there's anything else, Venkatesh. So if there's more questions on this week's topics or what we just discussed, please bring that to my attention. Otherwise, we can close down the session. All right, then. Thank you, people. Enjoy your weekend.